and thanks everyone for coming. And thanks to Louise Law, who originally suggested this as an idea. So just a bit about my background. I did an archaeological science degree at Sheffield and sort of got into ancient DNA when I got a postdoc at the Natural History Museum on a big sort of British-wide ancient genomics project. And when I began it, I really knew next to nothing about ancient DNA research. So I've really only developed an understanding of sort of how the field has progressed in the last few years through working with geneticists at the Natural History Museum and elsewhere. And so I'm still obviously not a geneticist, but I feel like I have a good, at least, conception of what potentially archaeologists should need to know and don't need to know about genetics. So I'm going to give what is probably going to be quite an intense description through what I feel like archaeologists need to know in terms of looking at ancient genomics and sort of understanding the kind of nuts and bolts of it. And there's quite a lot of stuff here, so I apologize if it's intense, and it's probably a good reason why it's being recorded, in that if you need to ever go back to any aspect of it, hopefully that you can. But I mean, there's some parts of this that are totally oversimplified, some parts that are completely beyond mine, Ken. But are kind of enough that you need to know to hopefully be able to scrutinize ancient DNA research in the future. So I've just put down a basic structure of this talk to make it a bit more coherent. I'm just going to go through some really basic stuff about DNA with you all to begin with. And some of this, many of you probably already know, but just to kind of catch up everybody and have everyone at the same level, then talk about what's changed in the last 10 years or so, which has meant that ancient DNA has become much more of a thing than it was before. And then start talking about more practical things, particularly going through metrics of ancient DNA quality, what information we can get from ancient DNA and how we get it. A specific section about genetic ancestry and particularly population genetics. And then I'll run through an example of a typical set of analyses that people do on ancient DNA papers and explaining what they're trying to do and what they use to find out. So beginning with the basics. So as probably all of you know, you can think of DNA as a sequence of letters G, C, A, T, with each letter representing a nucleotide, cytosine, guanine, adenine, and thymine. And the sequence of nucleotides is structured in this double helix structure, and you have with each nucleotide forms a base pair. So you can see in this diagram here with another nucleotide within this double helix structure. So G forms bonds with C and A with T. So together, those two nucleotides together are known as a base pair. And the human genome contains 3 billion base pairs around that altogether. In humans, these sequences of DNA are organized into pairs of chromosomes. One set you inherit from your mother, one set you inherit from your father. And you have 22 pairs of chromosomes plus the sex chromosomes, the X and Y chromosomes. And a minority of people have extra X or Y chromosomes, what are called intersex conditions. And actually, ancient DNA has identified a couple of people already who, in the past, who had intersex conditions. And these chromosomes are found in the nucleus of the cell, so are often referred to as nuclear DNA. You also have the mitochondrial genome, which is almost like an extra chromosome. It's just a genome in of itself, and it's just by itself. It's not in a pair. And this is found rather than the nucleus, it's found in the cell mitochondria, which are these little organisms that pack into a cell and help to generate the power. And this is inherited exclusively maternally. So your mitochondrial genome will be more or less almost completely the same as your mother's, but you have a different mitochondrial genome from your father. And recombination. So during reproduction, so you have all your set of your pairs of chromosomes, and they recombine together. So they are all mixed up together, and then one of those mixed up chromosomes gets passed on to your child, one from each parent. So you can see these diagrams here. You have someone with a pair of a blue and a red chromosome. They get recombined. and then they inherit one, which is blue and red, off their father. Similarly, the mother has a green and a purple chromosome. They recombine, and they inherit another off their mother. So 50% of your sequences, more or less, it varies depending on the individual because recombination is quite random, but it's around 50% from each. And the mitochondrial genome and also the Y chromosome do not recombine. The mitochondrial genome, as I said, because it's by itself, there's nothing to recombine with. Similarly, the Y chromosome in males, they only ever have one, so again, it can't recombine with anything, so it gets passed on. The only way that they change is through mutations. So every few generations, you get every generation there is some mutation that occurs in all parts of the genome. And because they don't recombine, and because you can obviously only inherit a Y chromosome from a male from your father, usually, different they've been used to track maternal and paternal lineages, and they're classified into things called haplogroups that have these funny long combinations of letters and numbers. And they were often used in older studies and still used today to some extent. They are still useful of ancient and modern genomes to look at past population histories by looking at distributions of paternal lineages and maternal lineages. But it's always good to bear in mind with these that they represent only a tiny proportion of an individual's overall ancestry, and is extremely biased towards male ancestors on one hand for the Y chromosome, and the female ancestors with the mitochondrial genome. And mutations. So when the DNA of your mother and father is recombined and copied, it's never done perfectly, and there's always some mutations where you have one nucleotide that is one thing in your mother or your father, but it's different with yours. It changes. It's sort of an error in the copy. Most mutations that occur are neutral and have no real effect, but the mutations in functional parts of the genome, which I'll talk about in a minute, create a landscape of variation, and that's what natural selection acts upon. So genes, as much as genes are a big thing that's discussed in genetics, most of your genome is not comprised of genes. So genes is something that we know is a sequence of these nucleotides that we know that encodes proteins. So it's directly functional. We know it has a direct function. And these genes aren't necessarily nucleotides all packed together in one long line. They can sometimes be distributed over different parts of the genome. And different versions of genes are called alleles. And like I said, 92 to 85% of the genome are not genes, so they don't have a direct functional effect, although some genetic variants outside of genes may have an indirect function, as we'll talk about later. So the thing that you'll hear probably archaeogeneticists talking about the most are these single nucleotide polymorphisms, so pronounced SNPs. And a single nucleotide polymorphism is a nucleotide position that we know commonly varies between individuals. So we know that is a position. So we know that is a position in which two individuals can vary in terms of what nucleotide is at this particular position, whether it's a G, an A, or a C, or a T, and it can change. As I mentioned earlier, we have about 3 billion base pairs of DNA, so 3 billion nucleotides. And tens of millions of these are SNPs. So tens of millions of these we know can vary between individuals. So that's basically 99% of nucleotide positions are similar between humans. So this is one of the metrics. There are various different kinds depending on what kind of genetic variation you're talking about. But this is one of the metrics that's rolled out in discussions of the fact that humans are 99% similar. But it's also true that tens of millions of SNPs, that basically gives you tens of millions of points of data where humans can vary and can be used to sort of analyze populations. So that's an extremely large amount of data, and it produces, even though it's only a small proportion of an overall genome, it's still extremely powerful. So the fact that we can distinguish, we can look at genetics of populations and look at differences between them isn't testament to any kind of profound difference between human populations. Human populations are very similar, and humans generally are genetically very homogeneous in general. It's more the fact of just how rich the genome is as a data source, that even when you look at a tiny proportion, you're still looking at millions of points of data. And also it's to do with the power of the statistical techniques that have been developed to look at these things. So SNPs are sometimes within genes, sometimes most are outside genes, and can have an effect within genes, but can also have an effect outside genes. So perhaps an indirect effect or regulatory effects. They don't produce proteins themselves, but can sort of have some role in how genes or other actual functional parts of the genome produce proteins. And like genes, different versions of SNPs, different variations on those positions of SNPs are called alleles. So when is DNA ancient? Technically, we should really call it, in some respects, should we call it ancient DNA? It's damaged DNA. Anything that utilizes damaged DNA is usually using ancient DNA, what we call ancient DNA techniques. So basically, the DNA after death very quickly fragments into sequences that are maybe on average around 35 to 40 base pairs long, and this occurs very early on. So that's the main thing, is that they're fragmented, also that they're damaged. So what happens is that these DNA fragments, and then over time that you get chemical interactions with the ends of the DNA sequence causes this damage. The damage isn't just on the ends, it's on the internal bit as well, but it mostly occurs on the ends because they're the most exposed. So when the DNA breaks up early post-mortem, you have just this soup of DNA sequences all fragmented. And the damage of the DNA sequences is related to time, temperature, and precipitation in terms of how the damage progresses. And basically it's looking to some extent in having this average 35 to 40 base pairs long, because when sequences start getting smaller than that, then they become too small to be able to do anything, be able to do anything with. So they become very non-specific because you only have a few of those nucleotide letters to be able to compare to anything. So moving on now to what has changed in the last 10 or 15 years or so, which means that ancient DNA has become so ubiquitous. So the old way we used to look at ancient DNA was using polymerase chain reaction and Sanger sequencing. So what you did essentially was you sampled a bone, and you used these DNA sequences called primers or baits to essentially fish out predefined sequences. So you say, I want to look at this sequence of the genome. You design a particular bait to capture that bit of genome out of your sample, and you put it in and you catch it, and then you use this method called PCR to essentially clone that sequence many times over in order to get it up to a point where the amount of DNA that you have is large enough to be able to then sequence in a process called Sanger sequencing. So for instance, you would target a particular part of the mitochondrial DNA that you know would tell you something about an ancient individual's maternal genome. So obviously from this, you're only looking at sequences that are maybe hundreds to thousands of base pair long, so a tiny fraction of the overall genome. So you're looking, you tend to predefine and look for small parts of the genome that you know will be particularly informative. What the issue is with these techniques, particularly for human DNA, is that it's very difficult to control for contamination. It's always difficult to be absolutely sure that you haven't accidentally amplified a bit of DNA that from somebody who handled the bones, or from even yourself, from a skin cell or something that just landed on the bone while you were sampling it. Because what these methods often do is preferentially actually amplify the contaminant sequence over ancient DNA. So you were never totally sure whether the result that you got through these methods was real or contamination. Now, what we can do now, the big technological change that's facilitated these new analyses is next generation shotgun sequencing. And by shotgun, we mean just sort of like a random scatter. So this is high throughput. It's quicker and cheaper to just get to sequence lots and lots of base pairs, basically. And in terms of the random scatter, what it means is that you just you have your sample of DNA and you just sequence a random selection of the DNA fragments in a sample. So you're looking at hundreds of thousands to millions of sequences distributed not just at specific parts that you've predefined, but across the entire genome, across all 23 pairs of chromosomes, but also you get the mitochondrial DNA and Y chromosome. And one of the main things that this allows you to do is that you can also authenticate ancient sequences. So you can look for that DNA damage, which shows that sequences are genuinely ancient, which you couldn't do before. So we can be much more sure of getting genuine ancient sequences. So this procedure, this new procedure, there's several stages to it, and it produces particular products, which is good to know about, because one thing this new ancient DNA techniques do is produce a lot of legacy products and legacy data. So basically you start off with a bone powder and you perform a DNA extraction, which is exactly what it sounds like. You extract the DNA from the sample. The DNA extracts are a finite resource, so eventually you will run out of that. But there's often DNA extracts left over from DNA research and which are stored and which people can access after the event and can be made available to future researchers rather than having to go back to the original sample. There's then a process which is called a library build, which is essentially where you, again, use PCR to clone your DNA extracts up to a level where they can be sequenced. What you also do is index the sequences as well, where essentially you fit little, in the laboratory, fit little tags to each sequence to associate them with a particular sample so they don't get mixed up with other samples. And this is called a DNA library, and these libraries are essentially immortal. If you start running out of a DNA library, you can just clone it up again and get some more. So again, this is essentially more or less a permanent resource that people can use without having to go back and sampling bone. And that's when you sequence the DNA. So you sequence all of the a random scatter of DNA fragments from your sample. And what this does is you can see here, so and then a computer program aligns these DNA fragments to the human reference genome. So if you can see this sort of diagram, you've got here what is the human reference genome, and then all of this stuff underneath are sequence reads of DNA, also sequences of DNA that have been sequenced, and then they get aligned to the genome, and you basically have different sequences which align to the same parts of the genome. So you get this kind of Tetris effect where you've got a sequence here, a sequence here, a sequence here, all stacked on top of each other. And the more you sequence, the more sequences you get back, and the more stacked that these sort of lengths, these more stacked, these sort of little these regions of the genome get. So basically you do shallow screening, you only get a few reads that cover in these parts of the genome. More and more sequencing, you get more, it gets more and more stacked. And sequencing data is always uploaded to publicly accessible repositories on publication. So essentially this data can be mined in perpetuity, and people can go back to the data before they go back to sampling, before they even go back to extracts and libraries. So hopefully once something's been sequenced, it won't have to be, people won't have to go back to it for a long time afterward until the techniques significantly improve. There's also, in parallel with this method, targeted in solution capture array. And what this does is you essentially, a bit like PCR, design baits to enrich samples for predefined human sequences. So you say, I want to look at all of these sequences in the human genome because they're particularly interesting. And then you chemically sort of enrich your sample for those. So think of it a bit like panning for gold. You catch all of the, well, not all of the sequences that you're looking for, but you attempt to capture all of these particularly informative sequences, and everything else is sort of washed away. So you get rid of all the non-human DNA, and you target sort of these large numbers of variants. So the common one that is used is the 1240K SNP, so 1,240,000 SNP capture array, where it's designed around capturing these 1,240,000 SNPs. And this is useful in one sense because it gives you the same set of or a more consistent set of sequences, rather than being a bit random all over the genome. You have these particular set sequences that you're more likely to get. And the vast majority of human DNA data has been that's been acquired so far has been generated this way. But the problem we've targeted in solution capture array, is that it only represents a fraction of the genome, a fraction of potential information that you want. And you, as well as losing any non-human DNA, you also lose human sequences which aren't on your capture array, which you haven't predefined. So there could be interesting genetic variation that we haven't yet identified that could be lost from using this capture array method. So the data legacy of this stuff is poorer because you can only ever get those predefined variants from it. You can't get variants that you can then that then might become interesting in the future. And again, there's problems with it representing a fraction of a genome. Also, a lot of the parts of the genome that have been already predefined have come from studies of European populations, and so using this capture array on non-European populations means it is less effective as well. So there's an ascertainment bias. But often, if a sample's poorly preserved, there just won't be a decent level of information inside that sample to get for instance, genetic sex, ancestry, and relationships with other individuals out. There just isn't enough. It's just not economical to shotgun it, because when you're shotgun sequencing it, you are just you're not getting enough information back. So a capture array method is the only real way of getting an adequate amount of information out of very poorly preserved samples, rather than using a shotgun approach. So the optimal approach is that when you have a really well preserved sample, you use shotgun, or a decently preserved sample, you use shotgun sequencing, where you just get random selection of the whole genome. When a bone is very poorly preserved, a capture approach is best, because you're never going to be able to shotgun that up to a level without spending too much money. So it's horses for courses. Okay, moving on to measurements of data quality. So one of the things, again, that you'll hear a lot of people talking about, and it comes up a lot, is the endogenous content. So this is the proportion of sequences that you get out of your sample that align to the human reference genome. So some of the very early ancient DNA studies that were done using shotgun sequencing were finding that basically less than 10%, sometimes even like 1 or 2% of their samples actually were endogenous, came from human DNA, and the rest were sort of bacteria, mainly from the environment, or microorganisms from the environment that had been living inside the bone. Although interestingly, now we know that within this sort of soup of microbial DNA, you also have pathogen DNA that were infecting the person when they died. So a lot of new studies that have come out recently have been looking at ancient pathogens. But what changed very quickly was that people realized that sampling of teeth, the petrous portion of the temporal bone, which the temporal bone is the bone around your ear, and the petrous part is this pyramidal aspect that surrounds your ear canal. And more recently, ear ossicles, the tiny bones of the inner ear, retain very high levels of endogenous DNA, not exclusively, but more like, are certainly more likely to have very high endogenous contents. So it's variable, but sometimes you get petruses and ear ossicles that have up to about 90% ancient human DNA in them. So one of the next measures of sort of the quality of a sample is complexity. So complexity represents proportion of unique DNA sequences in a sample. So essentially, because of breakdown in all DNA of the DNA, in all DNA samples, there is only ever a finite number of unique sequences in there, and that might not cover certainly probably won't cover the entire genome, and may not even cover the bits that you are particularly interested. And when you're shotgun sequencing, always unique sequences plateau eventually. So you get no new information. So if you're following this curve, you might sequence a sample once, you're getting pretty much all unique sequences. You sample it again, you sequence it again, then start your unique sequences start to drop. So you're just getting the same sequences back again and again, what are called duplicates. So which aren't giving you any more information about the sample. And this happens with all ancient samples. They are not an infinite source of information. The information starts to drop off after a while. This one here is the ideal situation where you're just getting a constant stream of unique sequences all the time, whereas this is what happens eventually with all ancient samples. The amount of unique information you're getting each time starts to tail off. And so again, this goes back to the capture approach as well. In DNA libraries that have low complexity, again, complexity is associated with general DNA preservation. It's very difficult sometimes to get an adequate level of unique information. So essentially to get enough information to look at things like genetic sex, ancestry, and relationships with other individuals. And so targeted capture analysis can salvage poorly preserved samples that have low complexity, because essentially it just grabs those bits. Targeted capture approach just grabs those bits that are useful and washes away everything else, and you're just sequencing those useful bits, even though it's only a partial part of someone's genome. And as I said, estimating contamination has become sort of standard practice now, and there's various different ways that are doing it to kind of make sure that a sample is not contaminated. So there's always some contamination, so in small amounts, so DNA of other human individuals in there, but this is nowhere near as much of a problem as previously thought. Contamination is only usually an issue when the ancient DNA preservation is so low that it's essentially lower than or equal to that baseline level of human contamination, that tiny baseline level of human contamination. So a lot of the time when we find contamination, it's actually just that the sample is poorly preserved. So as I mentioned before, one of the ways we can do this is looking at DNA damage on the end of fragments of DNA and authenticating sequences that way. We can also look at the mitochondrial DNA. So we know that essentially, because the mitochondrial DNA, there's only one of them, there isn't a pair. We know you can only get one set of nucleotide sequences, whereas other chromosomes, because there are two of them, you can potentially get different alleles on different chromosomes at the same position. So with the mitochondrial DNA, if we're constantly getting different alleles showing up in the same positions on the mitochondrial DNA, we know there's a second individual present or a second mitochondrial genome present. So there's some contamination. You could do the same thing for the X chromosome in males, because they only have one X chromosome. So again, we would expect to get only one nucleotide consistently turning up as being in that position on the X chromosome. If we're more likely getting sort of two possibilities showing up or more on the Y chromosome in males, and it suggests that there's a presence of X chromosome. Another X chromosome is there, and so there's some contamination. So what this means is that we can control for contamination, which is good, but also the results we've had so far suggest that it's not a big problem. So the idea that was touted a lot in the notice about archaeologists excavating hazmat suits in order to prevent DNA contamination, that's no longer a problem. I mean, we prefer people to be as clean as possible, obviously. But even if they're not, we've sequenced stuff that's been handled thousands of times, and there's very little contamination. So going back to this here figure here, to the idea of coverage. So again, this is a measure of data quality. So as I was saying before, shotgun sequencing produces these stacks of sequences that lie over the same part of the human genome. And coverage usually refers to the average number of unique sequences that cover a nucleotide position. So this nucleotide position here is covered, say, 1, 2, 3, 4, 5, 6, 7, 8, all the way down. So you basically produce an estimate of, on average, what's the coverage, like a number of sequences that are covering a particular part, a particular position within the genome, a particular nucleotide. And so essentially it's reflecting data quality and as it says, the coverage of the genome, how much of the genome is covered particularly well. And it's expressed as 1x, 2x. So 1x is each nucleotide position is covered on average once twice, 3 times, etc. And coverage is important because of the nature of the diploid human genome, that we have 2 chromosomes. Because you can have you have 2 chromosomes, you can have different alleles, so different SNPs in the same position on the different chromosomes. But because many analyses are performed, many analyses, they're often just looking generally at similarity and difference in the genome. They essentially just assume that the human genome is what's called haploid, so where you just have single chromosomes. This is what's called a pseudo haploid sequence. You just pretend that there's only one set of chromosomes just to get a consensus sequence to compare. And this is fine for looking at these metrics that are related to how similar or genetically people are, because you're doing essentially the same thing to everybody. It's more difficult treating, and that produces this consensus sequence where you basically compress all of the sequences down into a consensus of what nucleotide is present at a particular position, and just flatten it into a single consensus sequence. Using sort of haploid or pseudo-haploid genomes is more difficult for looking at phenotype, levels of inbreeding, population size, and selection, where natural selection, where you need to know the states of the alleles on both of the chromosomes. So the way this is done is sort of probabilistically. So if you have a 1X, you only have one sequence covering a particular nucleotide, you can't know whether sort of the nucleotide on the other chromosome is different. If you have sort of 2X, and it's the same, this comes down now to probabilities. So the higher the coverage, if you have sort of 10X and 5 of the positions in those sequences are one allele and 5 are another allele, you can be pretty sure that one chromosome has one version of the SNP, the other chromosome has another, because it's 50-50. So this is how coverage becomes important, in that the higher the coverage that you have, the more confident you can be about sites that are heterozygous or homozygous. So either having different versions of the SNP or different alleles on the 2 chromosomes, or whether they have the same alleles on the 2 chromosomes. And for that, you need high coverage. So moving on to what we can find out in the information that we get. The main thing you often get back initially is that we run a shallow screening run of the DNA library. So we take the library and we just produce a little bit of data from it. And sometimes this involves using a small capture array to see how well a capture would work on this library too. This is essentially to assess whether we can shotgun sequence it further and get sort of a full detailed genome, or whether we need to do some capture to get more data out, or whether there's no point in continuing at all. So we have an assessment of, so this is mapped over 35 is the endogenous content. Duplication, so this is how many duplicate reads rather than unique information we're getting back. The C to T is the damage, so we expect that to be 10% or over for ancient sequences. And you get complexity, a measure of complexity, so the unique sequences in the genome, how much you need to sequence it before it starts plateauing and you stop getting unique information back. And often with these screening runs, you produce enough data to produce information on genetic sex, very broad ancestry, and any close familial relationships with other sampled individuals. But some samples will also fail at this point, although fail is usually context specific, that in terms of the economy and the techniques available to now, we can't really produce any more data from it. But certainly people can go back to that library in future when maybe sequencing is cheaper, or more can be done to get more genetic information out of these samples, and do it then. So and do some more with it. So potentially, in a lot of cases, things that are classified as failure, people can potentially do something with in the future, and without having to go back to the sampling, destructively sampling the bone. So genetic sex is what we're really talking about is karyotypic sex here. So the presence of X and Y chromosomes. So essentially what you're doing is you look at how many of your reads or sequences align to the X and Y chromosome, and it produces a ratio. So XY would predict generally males have an X and a Y, and females have an X and an X, two X's. So you would get lots of just as many reads aligning to the X as the Y chromosome if it's likely to be more likely to be a male individual, and for a female you wouldn't get, or you get very few reads aligning to the X chromosomes. And as I mentioned, you can also potentially pick up intersex conditions where you get XXY or XXYY with this technique as well. And looking at genetic relatedness, so this is mainly based on percentage of DNA shared. So first degree relatives, siblings, or father or parent's child, they're going to share around 50% of the DNA. So they'll be 50% similar. 12 to 25% is second or third degree. So 25% would be sort of uncle or grandparent and grandchild or nephew. Third degree is sort of cousin or great grandparent. And you can combine this information with radiocarbon dates and uniparental haplogroups to work out the sort of direction of these relationships. But newer methods that are looking in more detail are starting to identify, and this will be the future of DNA studies in the future, particularly looking at migrations that can detect fifth to sixth cousins. So looking at very distant relatives distributed over different parts of the world, ancient relatives, can help figure out how far people were moving in sort of a certain, in generations, in generational timescale. So the last thing that you can talk about is genetic ancestry, which is here a whole topic in of itself. So what we're really trying to get at with genetic ancestry, the sort of core, is what's called the ancestral recombination graph. So this is a map of every bit of DNA that you've inherited of every ancestor that you've inherited DNA of, and you can track those and being able to track this bit of DNA that came down through, say, your great-great-grandfather to your great-grandfather to your grandfather, and watching how it goes, like sort of passes through all of your genetic ancestors into you. We can't get at the ARG without essentially sequencing most people who have ever existed, or at least a good proportion of them. So what we do is investigate ARG-like structures instead, and the most common is allele sharing, so in non-functional SNPs. So we look at SNPs that are, we know seem to be, or are largely going to be neutral, and the reason we look at non-functional SNPs particularly is because functional SNPs can be affected much more heavily by natural selection. So if they're doing something, any changes in the frequencies of alleles of those SNPs could be due to natural selection and adaptation at the genome level. Whereas in the non-functional SNPs, which are neutral, we know that any change has to be related to sort of population history rather than selection. And allele sharing, and why allele sharing is useful here is because humans mate non-randomly. And so things like geography, basically people who are closer together tend to more often have children together. People who are topography, so large mountain ranges, large seas often act as barriers between gene flow, between people having children together. And sometimes culture with things like endogamy can affect who people have children with over generations. And what this does is affect the similarity of people's genetic ancestors, so unshared genetic ancestors. And so this affects the alleles, so those versions of SNPs that these individuals and groups have in common. And so the sort of non-randomness of human reproduction means that you get nodes and structures that appear in genetic ancestry and allele sharing. So you have groups that share more alleles than with each other than others do, although this is always a clinal thing, and there's always a cline in terms of allele sharing between populations that are near one another, and then the cline is affected by these geography, topography, and culture. And it's also worth saying that structure is present at all resolutions. So essentially you could keep on finding structure in your data and allele sharing until you got down to essentially families. And they could go from families to extended families to groups like this. So there's no resolution at which you are at. This is the structure that defines humanity. The structure is always there, down to sort of the highest resolution level. It's just whether we can detect it or not, and whether it's useful, whether looking at a particular type of structure is useful. And it's important to bring up the fact that the history of genetics, and particularly human classification, which genetics sort of sprang off, is steeped in racism and developed in the context alongside eugenics in the context of colonialism. And a lot of the statistical tests that were devised to look at genetics and that still sometimes form the core of genetics today by people like Francis Galton, still are at the basis of the statistical tests that we use today to look at population genetics. And he was essentially using this in order to prop up hierarchies of racial classification that developed out of colonialism. And population genetics is still grappling with these legacies and sort of accusations of essentially trying to develop some kind of scientific racism. And there are people who manipulate and misuse population genetics to try and support racist ideologies. And so we always have to be careful in how we communicate this and try and fight this back. But as I was saying earlier, genetics of human populations are always clienal. Any categorization that we use is always arbitrary and is context specific, so that we group populations and individuals together in order to answer questions. But in that grouping, we are not saying that this group is anything meaningful outside of the study that we're performing. And it's important to say on that categorical level, race particularly is defined socially. It's not defined by genetics, and is context specific. So how you define racial categories changes in wherever you are in the world and the society that you live in. And also, race is a wholly inadequate way of categorizing human genetic variation anyway. So 2/3 of human genetic variation is within populations who live in Africa. So if you were to try to use genetics to divide people at kind of a high level, you would essentially have several populations in Africa and everybody else. So you can see in this right-hand side, the genetics of, I mean, this is a very simplified diagram, but the genetics out of everybody outside, who have ancestry from outside Africa, essentially sits within African genetic variation. Every human populations from outside Africa are essentially just one part of or fit within the variation of the genetics of a specific populations from East Africa. We're kind of just one offshoot from genetically from populations from East Africa. So when we define sort of populations and ancestry in AAD, they are defined in different ways depending on the context. So it's in part usually informed by genetic structure we see, but also time period, geography, and archaeology. So if we have a group of people who lived in a particular place at a particular time, we might just draw a ring around them as a population to compare to, to compare to other populations, to try and investigate population histories and admixture and gene flow. We're not saying that they are, and it's very rarely the case that they are a bounded, discrete, specific population. It's just useful to treat them that way, as in a modeled way, to ask some questions. But obviously, it's very difficult. That doesn't always come across, and the way that geneticists talk about populations definitely gives the wrong, in a casual way, or for the sake of convenience, in the way that we talk about them in a simplified way, means that people do get the wrong idea. And there are some geneticists who are trying to sort of rectify that. And in this context, it's important to talk about when human geneticists talk about modern populations. So in ancient DNA, we more often use sort of modern population data from modern populations to sort of compare it to Asian populations, to see whether we more or less have the full story of the genetic history of a place, whether there are any significant, where it looks like there are any significant changes in the genetic ancestry of people between a point in the past and the present. And so what we often want to do is try and get a sense of the population that existed in a place before more recent modern periods of well-documented migration. And the way that people usually do this is to sample people whose grandparents or great-grandparents are documented to all come from the same place. So it's to try as far as possible. I mean, it's probably not successful to get past more recent periods of migration that are well documented. But what they also do is that always, even with this very specific subsample of a population who lives in a place, and it is a very specific subsample, it's not in any way saying that these are the real people who occupy this place. There's then a removal of outliers. So you always get even genetic outliers, even when you've got this sort of conservative definition, this conservative grouping of people who have a kind of longer ancestral legacy in a particular place. You still get outliers that don't fit into the pattern. So they're often removed, and it's common that around 15% of the people who are included in a group initially are removed because there are genetic outliers. So these do produce groups that are, which are very conservative groupings of genetic variation in the past, but they are useful. We know that they're artificial model populations, but they are useful to be able to compare again to these ancient populations and see whether there's any difference. You know, I mean, eventually we won't need to use them because hopefully we'll have genetic sequences from populations all the way up to the present day, but it's definitely, we know they are not representative of the population of that place. They're just a useful model. But I mean, again, often people misunderstand this when reading the papers and don't have that background, because people use shorthands of these populations like Europeans, which geneticists are becoming more appreciative of why calling a group of people Europeans, a genetic cluster of people Europeans, is becoming more problematic, in that you're defining these as Europeans, and anybody who has ancestors that sort of arrived in Europe more recently are not Europeans. And people are coming to terms with that, but are trying to grapple with those issues and try and come up with a better, more nuanced way of discussing these things that for convenience sake are discussed very simply in the papers, but often get misconstrued. And one of the big things I always to remember, particularly when communicating this with the public, is that genetic ancestry is not genealogy. You've not inherited, you have no DNA from most of your ancestors. So you basically inherit DNA in chunks. And when you go back a certain amount of time, you have more ancestors than you do inherit chunks of DNA. So eventually you have ancestors that drop out that you have no DNA from. And even when you get to 10 generations back, the chance that you've inherited any DNA from any one ancestor is very small. So if you look at this graph here, this is looking at a person living today, 17 generations back. The blue, so the whole thing is their number of genealogical ancestors, and the blue is the proportion that they will have inherited DNA from. So it gets to a tiny proportion, and also you can see that as you go further back in time, your genealogical ancestors spread out exponentially across geographic regions. So will be from all over wide geographic areas. But because most of your ancestors, your genealogical ancestors, will be from a particular place because you were born in that particular place, the probability is that you've inherited the majority, you've inherited the majority of your DNA from the ancestors that lived in the same place as you. So a modern individual's genetic ancestry is very much dependent on their recent ancestors, so the ancestors they're more likely to have inherited DNA from. And so it reflects more broadly, and it's kind of nested as that Russian doll effect. And then those people are more likely to have inherited DNA from people who recent ancestors lived in that area, and it kind of goes back and back and back. So what you get in your genetic ancestry is the population history of the area where your very recent ancestors came from. It does not connect you to, in a geological sense, to ancient people in a meaningful way. And as well as this, the number linked to this, so as you go back in time, the number of your ancestors is increasing exponentially, but the total population is decreasing. So you get pedigree collapse, which is where your multiple lines of descent pass through the same historic person. And then eventually you get to this the genetic isopoint, where essentially everyone who existed in a particular area at a time in history is the genealogical ancestor of everybody with ancestry from that area who exists today. So basically everybody who, this is called the genetic isopoint, and the genetic isopoint for Europe is the 10th century AD. So basically if you have any kind of recent ancestry in Europe, everybody who was alive in Europe, in any part of Europe in the 10th century AD, is your genealogical ancestor. We all have the same ancestors in Europe in the 10th century, that were in the 10th century AD. It's a very counterintuitive concept, but it's sort of been shown to be true. So again, pointing out that your genealogical ancestor is not your genetic ancestry, and these are two different things. So when we go back to someone like Cheddar Man, who lived 10,000 years ago, he lived so far back in the past that if he did successfully produce descendants that survived in the present day, he would essentially be the genealogical ancestor of more or less everybody alive on Earth today. And similarly, isopoints for the whole world are somewhere, or not so far back, somewhere around the 3rd millennium BC. So essentially, everybody alive today has the exact same ancestors distributed all around the world in the 3rd millennium, the 3rd or 4th millennium BC. So moving on back to genome-wide data, the reason why it's so informative and so useful is that it provides data not just for an ancient individual, but you're also sampling a population of their genetic ancestors. And you're sampling them directly, genetic information from them directly. So essentially, you're not just getting genetic information from a single person, you're also getting genetic information of hundreds of thousands of people, hundreds to thousands of people whose remains may not have survived into the archaeological record. And this means that sample size tends to be less of an issue. So if you find one person who has a particular type of ancestry, ancient person who has a particular type of ancestry, then you can maybe say, this might be a recent migrant who's got ancestry from elsewhere. It only takes a few more individuals, because you're looking at their population of ancestors to start to turn up, to start to be able to conclude that this is actually a typical ancestry of people who are living in this place, at this area. What may be a bigger problem is sample bias. So if, for instance, there were only populations with a particular type of ancestry were burying their dead, and people with another type of ancestry were cremating their dead, and we can't get DNA from cremations, we would only see the ancestry of the people who were burying their dead, and so there would be archaeological bias there. But what we have to bear in mind with that is, for that to be absolute, there would have to be essentially the people cremating and the people burying could not be having children at all with one another, because as soon as they start having children, then we will start to see part of the ancestry in the people who are burying their dead. So there may be a bias in terms of the change that we see initially, but we should be able to tell if there is a population still there, unless we think that they are just not having children with each other whatsoever. And then also we can use modern populations to see whether any genetic change that we see in the past persists up until modern day. So if a population, the ancestry of a population is substantially diminished at a particular point in time, if that ancestry still isn't prevalent in populations that we see today, then we can sort of conclude that that is a persistent change in the genetics of people. So notwithstanding very complex, more complex situations of back and forth genetic change. So there are several different processes that can produce genetic change through time, and it's important to remember that genetics of human populations are always changing, although the rates vary. So our generation is different from the previous generation, it's different from the previous generation. We are not genetically same as people, even quite the same as people even 100 years ago, even though we might be very similar to them, generally speaking. And genetic change can occur through a variety of ways. You can have admixture, which is mixing the populations who are previously isolated from one another. Selection, so this is where a particular genetic variant, the phenotype of that variant, so what that variant does in the body becomes particularly advantageous in an environment, and so over generations becomes more common because it's advantageous. And you also have this process of drift, And this is change in prominence of particular variants due to sort of random chance and bottlenecking. So a good way to talk about this is that you have a population, and if there's a bottleneck, or if part of that population splits off and say starts a settlement over here, the frequencies of alleles in that population won't quite be the same as in the parent population. And the difference between those two, then if these two remain separate from one another, or if you have a bottleneck where the population, the frequency of the bottleneck has caused the frequency of the alleles in this population to change, then that population breeding amongst itself essentially exacerbates that genetic change. So variants just change and become more prominent in populations just due to random drift. This is called drift, just due to random chance. And a good example of this is in Iceland. So genetic analysis of early medieval populations from Iceland show that they're actually genetically more similar to populations who inhabit Britain and Ireland today than they are to people who inhabit Iceland today. And that's because when people from Britain, Ireland and Scandinavia went to Iceland, they had a very small population size, and so drifted very quickly. So they've essentially drifted because they're a small population, and that allows, and they were sort of taken as a subsample from these populations of Scandinavia and Britain and Ireland. They've totally drifted away from what they originally looked like, meaning that the modern people from Britain look more like ancient Icelandic people do than modern Icelandic people. So it can have a large effect at certain points. And it's important to define this concept of migration as is used in population genetics. And the problem is with population geneticists is that more often they're used to talking about migration to do with sort of reindeer and in those terms, rather than a lot of the inevitable social baggage that comes with talking about migration of humans. And the definition that they would use for migration here is people moving from one area to another and having children without any other aspects of push and pull factors and things like that, which, to be fair, they've been rightly criticized for in not considering that always when we talk about migration, we have to consider social factors as well. So any ancestry change that we see over time at a population level, so any change in ancestry that can't really be explained by drift, indicates that there must have been migration, people moving into this place and having children. And when often human genetics papers refer to mass migration, what they're often referring to is the size of the ancestry change that these migrations produce. So to me, and I've had conversations with geneticists about this in the past, that to me is not really most people's definition of mass migration, because they're talking about the large scale change in ancestry, not necessarily a large scale movement of people. And most of the time when they're talking about changes in ancestry, they're talking about, yes, large scale changes of ancestry, but occurring over hundreds, sometimes thousands of years. And so various mechanisms could be responsible. It could be that, for instance, a relatively small number of people move initially, but that they then produce more descendants in the long term than the people who were living there. So we're talking about, usually with migration, movements of people, and usually groups of people, that result in a very a change of ancestry at a population level. So it indelibly changes the kind of allele sharing in the population. And we can identify individual mobility too. So if you say you're picking up people with ancestry that's different from other people, but that you see no particular change in the overall ancestry in a particular place through time, that suggests that you're just getting odd individuals moving in, but not groups of people that are affecting change in ancestry at the population level. So I've now quickly, because it's 7 o'clock now, I'll just quickly run through typical types of analyses that people do when they detect migration, it as defined here in these sorts of genetic terms. So one of the first things people do is often this k-means cluster analysis, which is where you basically say split these samples up using this number of categories, and you define the number of categories that you want to split it up. And the categories are not important. What's important is how each of these populations or individuals behave when you have different K. So K is the number of categories. And you could basically keep on going, increasing K until you're essentially just getting families turning up together. So if you look at this plot, you can see here that K is 3 because you have 3 colors that are comprising these populations. And you can see that some are comprised of just, can be explained by 2 populations, some need 3. And you can see basically they are usually clustered in these plots in terms of their genetic similarity. And then also you can look at how they're colored to understand how each population are related to each other. So you can see that all these populations in Chalcolithic and early Bronze Age Britain all have this green component, which is absent from Scotland, Neolithic Scotland and Britain. So it suggests that there's a new component of ancestry that's been introduced in Chalcolithic and early Bronze Age Britain that wasn't there before. So this is look, this is just using one K, so K equals 3, but you can basically run through all K. So K equals 2, K equals 3. So you have the K equals 2, you have 2, the blue and the orange, and then you can keep on going up through K and you get similar samples behaving similarly. So you can see where your sample sits in the broad stretch of things. I know this is just psychedelic vomit, but you tend to just look at which K best illustrates similar behavior of your samples. You also have principal components analysis, which is where you essentially set out kind of a space of genetic variation, usually using modern samples, and you project your ancient samples onto it. What's important to remember about, and basically the samples cluster in terms of their genetic difference or similarity and allele sharing between each other. It's important to remember this is an exploratory task. It's not a test, and the relationships aren't always straightforward. So particularly you'll see that these axes only explain a tiny amount of variation, each component. So 0.82%, 0.37%, but it does give you some indication of the relationships between ancient individuals. So this from a paper I was involved with, where Mesolithic hunter-gatherers from Britain cluster with other Mesolithic populations from Europe, whereas Neolithic populations cluster with other Neolithic populations from Europe and are quite far apart, which indicates that genetically Neolithic populations of Britain were quite different, or the ones that we sampled were quite different from the Mesolithic populations of Britain, because they've shifted over here. But the relationships aren't straightforward from this PCA, because you have lots of different further dimensions. So it looks, for instance, like some of the Mesolithic populations of Europe are quite close here to modern populations of Europe. These are kind of grade, these fainter markers are kind of modern populations of Europe. But actually, when you go into kind of a third dimension that way, these are set quite far back from these modern populations. So you get a gist of the relationships between populations, but you don't get a sense of the distinct relationships between them. But it's useful for generating hypotheses. So what people then usually go on to is this thing called F3 stats, where again, you compare a population or an individual to another population or an individual and come up with just a quantify how genetically different or similar they are from each other. And these are often expressed in heat maps. So you can see that, for instance, Cheddar Man is similar genetically to, it's hotter, and so he's similar to a Mesolithic individual from a Welsh cave. And then when you get into Mesolithic populations from Scandinavia, it gets a bit colder because they form a slightly different genetic cluster to the individuals that are from Mesolithic Britain and western parts of mainland Europe. So again, you're just comparing individuals and looking at which populations are more similar, more different, and quantifying it. Once you've done that, you then use these F4 stats to say you have your target population or individual, and you say, genetically, are they more similar to this population, or are they genetically more similar to this population? And you do lots of these pairwise comparisons. So for instance, with and sort of this figure here, you have these two populations. This is comparing these different populations of Neolithic Britain and Ireland to Neolithic populations from Central Europe and Iberia. And if the value is positive, then it's more like this individual in the list. If it's more negative, then it's more like this individual here. And the Z-score indicates the significance of the result. If it's over 2, then it's a significant result. So you can see that this is showing that all of the Neolithic populations from Britain and Ireland are genetically more similar to, it's the other way around. Iberia is positive and Central Europe is negative. So because all of these results numbers are positive, all of these individuals are genetically more similar to populations who lived in Iberia during the early Neolithic than populations who lived in Central Europe during the early Neolithic. A lot of the times, it's very unlikely that you have the actual source population of any genetic change you see in a particular place. And no one sort of thinks that people were moving directly from these places into Britain at this time. But by comparing them, you can start to at least narrow down where populations who were involved in particular migrations came from, even if it's just a very vague region. So it's not to be taken too literally. Similarly, you have this process QP admixture, where you model an individual population as a combination of ancestry from one or more populations. So you start out with one. If that doesn't work or it isn't a good model fit, then you use 2 populations. If that doesn't work, you try 3. You don't usually really go much past 3 because it gets the model struggles to deal with it. And essentially, you can cycle through different populations from different parts of the world in different time periods and just essentially try them all to see what works. And again, it's not taken literally, so it's very unlikely in a lot of cases that you have the true source of any migration into a particular period. But it's just to say that if all of your samples or all of the models that fit are involving ancient individuals from sort of a particular part of the world, then that implicates probably that part of the world or somewhere around there as being involved in these migration events, even if it isn't very specific, or even if the samples that you're looking at aren't exactly in the same time, quite in the same time period that you're looking at. So for this example, we've modeled, ignore this top one, these bottom ones we've modeled Neolithic populations of Wales, Scotland, and England as combinations of people from the early Neolithic Iberia and Mesolithic Britain. So clearly people from early Neolithic Iberia are too old to be directly responsible for the early Neolithic in Britain, and they're also probably in the wrong place. But we can at least, they're a good fit for this population, so we can say that either Iberia is kind of a proximate source or an ultimate source of ancestry that comes into Britain. Yeah, so it's a proximate or indirect source for the ancestry that comes into Britain, even if they're not coming from Iberia itself. So the more ancient population data we get, the more we can narrow this down. But by testing lots of populations and in these models, we can look at broadly where they're likely to have been coming from, or at least narrow down the hypotheses of where population would like to be coming from when they migrated into different areas, and also the sort of magnitude of the change. So we can see that in Scotland, there's quite a substantial minority of the ancestry comes from the local Mesolithic populations. In England, it's lower, and in Wales, a lot lower, next to nothing. So you can use these models to try and quantify what the amount of genetic changes as well through these through these tests. So just a summary, and this is what you should expect if ever you submit a sample to an ancient DNA lab, and to some extent demand is, and going through all the things that I've discussed, whether a sample has, in their terms, success or failed, what they've done with the laboratory products, how people in future can access them, whether the data that they've generated was shotgun sequenced or using targeted capture or both, what was the endogenous content, what was the damage, the complexity, the coverage, all of these quality control indices, what genetic sex they got back, any information on the genetic ancestry, and any information on genetic relationships with other sampled individuals. In some cases, the DNA would not have been good enough to produce those metrics. but they should tell you if that's the case and tell you under what circumstances they failed. Also uniparental haplogroups, which can help, again, distinguish kinship relationships particularly, but also look at sort of population changes as well. And they should also give information as to where they've, when they publish the data, where they've uploaded their sequences and where people can publicly access them and use them again. And all of this is to ensure that we never, or not for a long, long time, ever have to go back to sampling the ancient bones again. And I'm sorry, I'm very sorry I've run over time. I'm sorry for sort of rushing a bit towards the end, but I'm happy to take any questions that any of you have about any of this.